Hey all, my name is Prashanth Kumar, or PK, and I'm an MCAT tutor at Shemmassian Academic Consulting. Today, we're going to be doing a passage walkthrough in the MCAT Biochem section. You should be taking roughly 6.5 minutes on this passage, but today, we're going to be going more in depth so you can see exactly what's going on in my head as I tackle this section. Let's get it. Alright, great. So I've gone ahead and put the passage on the left and the questions on the right. So feel free to take a moment, pause the video, and come back to us when you're ready. I'm going to go ahead and zoom in on the passage just because we got some comments saying it was hard to read, but we can just scroll through it slowly and pause the video whenever you need. Alright, and the questions. Awesome. And so pause the video, come back to us when you're ready. And with that, we can get right into talking strategy. For me, I like to maintain the same exact strategy coming into the passage as I do for every other paragraph passage section. The reason for this is it kind of boosts my confidence. I know that I'm going to come in with the same strategy. I know how I'm going to take the test, and that boosts my confidence. And confidence is super, super important on test day, having that plan of how you're going to tackle the section. My strategy is read the paragraph, write a quick 3 to 5 word summary, and then go on to the next paragraph, do the same, and on and on and on. The reason I like this is that it makes sure I understand exactly what's going on in the paragraph before I move on to the next one, sort of giving me a mental check-in before I continue on to the rest of the passage. The mental check-in is very, very important because oftentimes I'd find myself reading an entire passage and not understanding one part of it, and that messed up the understanding of the entire passage. So doing this little check-in makes sure that if I don't understand something, I can reread the paragraph much, much quicker than if I was rereading an entire passage, which would take up a lot of time. So this saves a lot of time and boosts our confidence as well. Next tip I have is I don't spend too much time on the figures on that first read. The reason for this is if a question asks us to come back to the figure, we'll know and we will come back to the figure. But if there's no questions being asked about the figures, why would we waste time going to it, right? The figures are often complicated and we don't want to waste too much time because time is a big concept in the MCAT. So with that, let's get right into this passage with my strategy. Postoperative acute kidney injury, AKI, is a severe complication after liver transplantation, as its deterioration and magnification can lead to an increase in mortality. Connexin 43, CX43, mediates direct transmission of intracellular signals between neighboring cells, always considered to be the potent biological basis of organ damage, deterioration, and magnification. All right, so the question I always ask myself after I read a paragraph is, what's happening, right? What is the big takeaway? If a paragraph had a thesis, what would that thesis be? What's happening? And I write it right next to it. So here, we're looking at AKI, right? Acute kidney injury. And we're using this arrow to show that C43 or CX43, this connexin protein, is the biological basis. It's like what's causing it. And so I just drew that arrow. And remember, this is your summary. No one else is going to be reading your summary on test day. So all that matters is that it makes sense to you and that it's quick to you. Use abbreviations, use symbols, use whatever you want. It's your test. And with that, we can go on to the next paragraph. Researchers obtained donor liver tissues of patients in order to explore the effects of CX43 on organ damage. Reperfusion is a biological process used to refill blood in AKI. Donor livers displayed more severe lobular distortion with necrosis, apparent edema, hemorrhage, and neutrophil infiltration after reperfusion, compared with donor liver tissues obtained before reperfusion or normal liver tissue from patients with hepatic hemangioma. After reperfusion, CX, mRNA, and protein levels were determined as shown in figure 1. And then it gives us a figure. Again, I'm not going to spend too much time on the figure right now, just looking at the x-axis, y-axis if I want to. It's talking about liver, pre and post reperfusion. So again, the question I'm asking myself is, what's happening in this paragraph, right? And then here, we're looking at reperfusion effect. I'll draw an arrow with a question mark on AKI, right? And so the effect is reperfusion is a biological process used to refill blood in AKI, and then there's two different effects, right? We're going to look at donor livers versus normal livers, right? And so I'm not going to write out all the details, right? There's a lot of details. I'm just using this as a roadmap. So if I need to come back to the paragraph, I will. And so you see I've written out a small little diagram to help me model this, right? We're going to look at reperfusion in donor and normal kidney livers. And if I see a question that talks about that, I know exactly where to come back to. And so here we also have a figure. And remember, I'm not going to spend too much time on the figures right now. But what I will do is see that the figure is talking about sort of these reperfusion before and after, as well as mRNA units. And so I see mRNA, I'm thinking transcription, translation in my head. I see a Western blot, I'm thinking translation. And so right away, I have all these ideas in my head, but I'm not spending any time like looking at the details of the figure too much. And we go right on to the next paragraph. Autologous orthotopic liver transplantation, ALT, rats were built by researchers in order to further study AKI. Researchers hypothesized that the change in CX43 expression might play an important role in AKI following ALT. Researchers used heptanol, a well-known inhibitor of CX43 without hepatotoxicity, to alter the function of junctions composed of CX43. Heptanol had no effects on CX43 expression in kidney or livers. Right. So the question is, what's happening here, right? And so here they're looking at sort of the effect of AOLT on AKI, right? And so how are they doing this? Well, they're looking at the CK43 mechanism, right? This or CX43. Where they're looking at CX43's effect in this in AOLT mice. And so how are they doing that? They're using heptanol to inhibit CX43. Okay? And so right away, that's like a big summary of what's happening, right? AOLT mice look at AKI, and they're using heptanol to inhibit CX43, an important mechanism in the pathway of AKI. And so that's our quick summary. And so here we have our first idea of like an experimental setup. And so next, heptanol or a sham control were injected intravenously into the rats, and the pathological score of kidney cells was determined to analyze disease progression. The results are shown in figure 2. And so then we have another figure. I'm not going to spend too much time giving a summary. We're just looking at hep versus sham on kidneys, right? That's all we're looking at, the effects of heptanol sham. And we see that the graph figure 2 shows exactly this, right? We're looking at some sort of score given by kidney function to these different mice groups. Researchers also believe that RIP1 may be involved in CX43 function, and they confirmed the interaction of the proteins through co-immunoprecipitation. RIP1 is a typical marker of necroptosis. All right, again, very short paragraph. So what's the question asking? What's happening, right? And so here, we're just looking at, does RIP1 involved in CX43, right? Question. That's how I'd write it. Just looking at this relationship between RIP1 and CX43, and RIP1 is a typical marker of necroptosis. So whatever you want to talk about, but I'm just writing RIP1 has a relationship. And if a question asks me about RIP1, I know exactly where to come back to. And so with that, we have a pretty good roadmap of exactly what's happening in this passage, right? We're talking about acute kidney injuries. We're looking at different experimental setups to investigate different mechanisms and proteins involved in acute kidney injury. And so with that, we can get right into our questions. All right, question number 1. What type of a junction does CX43 most likely form? Is it A, a gap junction, B, a tight junction, C, a desmosome, or D, a hemidesmosome? All right, so right away, we should be thinking about where does this come up in this passage, right? And so we're thinking CX43. When did we first talk about CX43 and what is it? So we come up here and we remember that CX43 is mentioned in our first paragraph. It's called connexin 43. And so what exactly is connexin? Well, connexin from our content review should be known as a type of gap junction, okay? Right? And so what exactly are these junctions, okay? So a gap junction is you have these two cells right here, and I am liking it to having a tunnel, okay? So they're channels that connect these two cells, And specifically, they allow them to share the cytoplasm of both cells. They're connecting the cytoplasms directly, so molecules, particles, whatever can pass through and connect the cell. Usually common seen with ions and gap junctions. Now, that is our answer because we know it, but let's make sure we understand what all the other ones are. So a tight junction, so I'll write gap, a tight junction is another type of connection, but I liken this one to glue, right? And so I'll put the glue in blue, okay? So it's like a glue between two cells that are neighboring. And what it does is it's like a water seal to tight seal, right? Kind of like Flex Seal. It keeps these two cells connected, but there's no leakage between the two. And so that is what a tight junction is, hence the word tight. A desmosome is a little bit different. It's a very, very strong connection between two cells, right? But instead of it being a glue in between the cells, I kind of liken it to sort of sewing, right? If you've ever sewn or you've ever stitched, it's kind of like a connection between these two cells, like that. And so it's kind of like tying a thread between the two cells, and the threads are attached to the cytoskeleton of both cells. So I'll draw it like let's use blue. It's like the cytoskeleton on both sides are tied together by these desmosomes, which are stitching them together. And a hemidesmosome is a slightly different type of attachment. So in this case, we were looking at cell-to-cell attachment in all three of these sections, cell to cell. But a hemidesmosome doesn't necessarily deal with the cell to cell. Instead, we have a membrane at the bottom that the hemidesmosome attaches the cell to. So if we draw it in red, it attaches the cell to this basement membrane, or this membrane, as opposed to attaching a cell to a cell. And so with that, we have a good understanding of all four of our types of junctions here. And so we should just know that connection is a gap junction, but this is really, really important and really useful to understand that all of these types of connections will come up on test. But great. With that, we're ready to go on to question number 2. Alrighty. Okay. A researcher discovers a new CX43 inhibitor made up of a glycerol backbone and 3 saturated fatty acid tails. Which of the following describes the path by which the inhibitor enters the cells? A. The inhibitor is recognized by a specific receptor. B. The inhibitor is taken up by a nonspecific receptor. C. The inhibitor diffuses through the cell membrane. Or D. The inhibitor passes through the cell membrane via a channel. All right, so if we have this CX43 inhibitor, the question is asking, how does it get into the cell? How does it enter the cell? And so right away, it gives us a descriptor of what the inhibitor looks like. And so it has a glycerol backbone and 3 saturated fatty acid tails, and that should be ringing bells immediately, right? If you have this backbone and you have 3 fatty acid tails, right? What do we call that? A triglyceride, right? Tri meaning 3, glycerol. And so here we have our triglyceride. And what is a staple important quality about a triglyceride? We should know that it is very hydrophobic, right? Very hydrophobic. Nonpolar, all of the above, right? It's very hydrophobic. And so how do triglycerides get through the cell, right? If they're nonpolar and hydrophobic, they are able to diffuse straight through the cell, right? Nonpolar molecules can get through the cell membrane without need of a receptor or a channel, right? And so with that, we can eliminate A, B, and D pretty immediately, right? Because they're talking about using a receptor or a nonspecific or specific receptor or a channel, which we don't need in any of this case since we're dealing with a very hydrophobic molecule. And so with that, C tells us that we have a triglyceride that's able to diffuse. So remember, nonpolar, diffuse through. Okay, great. On to question number 3. So that was another type of question where we don't need the passage information. So it's a standalone question, but it does require a good understanding beforehand. Awesome. Question number 3. researchers decide to interrupt the CX43 RIP1 interaction in the AOLT rat model without using a small molecule inhibitor. Which of the following will most likely disrupt the interaction? Is it A, siRNA for RIP1, B, radiation against heptanol, C, acetyltransferase specific for beta-actin, D, plasmid producing CX43? All right, so we're looking for something that is very likely to disrupt the interaction. And so in this case, we have 4 choices, but what are we talking about? Well, we're talking about RIP1, right? And so this is mentioned where? In that final paragraph, right? It says researchers also believe that RIP1 may be involved in CX43 function, and they confirmed the interaction of the proteins through co-immunoprecipitation, right? So since it tells us that it's a protein, we know that it goes through transcription, translation, all of that. And so now we realize that this is a protein-protein interaction. Great. And so basically it's asking how would we disrupt the interaction, right? So what we would do, right, is we're looking for an inhibitor. And so right away we can eliminate one that's pretty clear to not affect anything. And that's a plasmid producing CX43, right? Increasing the amount of CX43 does not necessarily inhibit it. We have no idea of what it will do, but generally having more of a protein will not increase the inhibition unless it's a specific type of inhibitor. So just because we have something producing it doesn't mean that that will inhibit it. Okay, let's go up from there. Acetyltransferase specific for beta-actin. We don't really hear anything talked about in terms of beta-actin. In fact, the only time we see beta-actin is generally in these Western blots, right? And why do we have beta-actin as a protein that's always mentioned? Well, this is something called positive control. It makes sure that we have the same amount of thing or same amount of substance being added to each of the wells in our Western blot. And so beta-actin is not going to be something we affect with an inhibitor, since it would not directly affect the CX43 RIP1 interaction. So we can eliminate C. So now we're down to the siRNA or silencing RNA for RIP1 or the radiation against heptanol. They both will affect RIP1 and probably the interaction. But the reason we're going to pick A over B is because siRNA for RIP1 is very, very specific, right? If we're trying to silence some sort of interaction or inhibit some sort of interaction, the best way to do it is to block the interaction directly between the two. And so here, if we have a silencing RNA, how does the silencing RNA works? Well, it works by attaching to the mRNA and sort of signaling some sort of degrading. So siRNA plus mRNA results in degrade, right? It's a signal to us for it to destroy the RNA. And so with that, we're going to pick A because that's going to get rid of the RIP1 and so thus cease the interaction between the two. Now, if we did radiation against heptanol, radiation is a much broader sort of protein. It'll disrupt everything, right? There's no guarantee that it will interrupt the CX43 and RIP1 interaction. But we can say that it is possible. But in this case, it's saying most likely disrupt, so we can eliminate B since A is pretty directly going to affect this interaction. And with that, we're 3 for 3. On to the final question. Great. Which conclusion about CX43 regulation is best supported by Figure 1? Is it A, all expression control occurs at the transcriptional level, B, all expression control occurs at the translational level, C, donor livers have higher levels of CX43 after reperfusion when compared to normal liver tissue, or D, normal livers treated with heptanol have lower levels of CX43 compared to donor livers treated with heptanol. All right, so right away, we know exactly where to look. Figure 1, right? And it's talking about CX43 regulation in Figure 1, okay? So we're just going to jump over there and look at what Figure 1's talking about. So Figure 1, we have two sorts of things, right? And I remember we talked about it briefly, but I always go with the axes. So right away, we see that we're looking at different types of liver tissue before reperfusion, after reperfusion, and we're looking at the mRNA involved in in each of these types of livers, right? And so we can see normal liver tissue is right here, and that there's generally a trend of before reperfusion and after reperfusion of increasing, right? And we see that this star on both of these shows statistical significance. And that's a very, very important term. Statistical significance is very, very, very important on the MCAT. It's going to be the differentiating factor of whether you can tell some sort of significant difference between two groups. If you don't see anything saying statistical significance, no stars, no brackets, whatever, you cannot say that there is a difference between two groups. And I want you to hold that with you as you take the test. And so we have that as our first diagram. The second part is a Western blot, right? So right here, we're looking at beta-actin levels and CX43. And so what does a Western blot show? Shows protein levels. That's very, very important. If you don't remember the mnemonic snowdrop, right? Snowdrop, W matches with P, so Western equals protein. Google this if you don't remember, but snowdrop is very, very important in telling you the different types of blotting. But in this case, we have a Western blot, and we are looking at protein levels, and specifically CX. So we see all of these diagrams, and we see that we're talking about mRNA levels as well as protein levels. And that is a general stacking, which talks about transcription, right, and translation. This is a common sort of staggering pattern where you see mRNA levels talking about transcription levels and protein levels as translation. So whenever we're quantifying protein levels, this is a very, very common sort of setup you'll see. But with that, let's go right into the question. Which conclusion about CX43 is best supported by figure 1? So right away, I'm actually going to eliminate A and B, right? All expression control occurs at the transcriptional level, and all expression control occurs at the translational level. The reason for this is it uses extreme terminology. Whenever you see all expression or all of something, you already know that's not true, because there's always going to be some sort of exception. And here, you can even see that there's changes in both transcriptional levels as well as translational levels, right? They're not identical in any case. The top one indicates transcription, the bottom one translation, so we can eliminate A and B immediately. Now C, donor livers have higher levels of CX43 after reperfusion when compared to normal liver tissue. So this is exactly what we saw, right? We saw that after reperfusion, compared to normal liver tissue or before reperfusion, we have the statistically significant difference, right? Indicated by that star, showing that there is a greater difference between the two. And so we can choose C as our answer pretty quickly. And then D, normal livers treated with heptanol have lower levels of CX43 compared to donor livers treated with heptanol, right? Where the diagram, the figure we were looking at, did not even mention anything about heptanol. So we're not even going to look at that. Remember, you're always looking at the exact sort of space they're talking about. Here, talking about figure 1. Is there any justification for this? No. And so we're left with C, and we're 4 for 4. We crushed it. All right, great work, team. We crushed another biochem passage. Kudos for you for getting it done. Congrats on getting through all that experimental design, understanding cellular structure, all of that. Good job. You crushed it. If you like this video, give us a like and subscribe to our channel. We really appreciate it. And comment exactly what you're looking forward to next and what type of walkthroughs you want to see. If you like this video, check out our 528 strategies series on our channel from Vikram. Super helpful tips and strategies to tackle all the different sections on the test. Also, click the link in our bio to sign up for the MCAT question of the day so you don't miss out on any test prep before test day. Happy studying, and let's get it.